Hello, everyone. My name is Austin Dobrecevich. Some of y'all might know me as Augie D, and I'm here to talk to you today about a senior project that I took on, which is working towards improving Culex tarsalis habitat suitability model that was existing. And so it all starts with this mosquito. Well, not this specific mosquito, but its species. And so this is a picture of a Culex tarsalis mosquito, which is one of about 50 mosquito species in Montana. And so this summer, I worked with several other students in trapping mosquitoes and then sorting through thousands of them to find this specific species. And so here are some key identification parts for IDing the specific species, and it has this rounded abdomen and also that white banded proboscis. And so those are things that we look for when sorting through the thousands of mosquitoes, and it's very unique to this species. And so this species of mosquito is found in areas that are plains with like cattle ponds. And so this is because they like what they call riparian vegetation. And this mosquito therefore exists in those areas in eastern Montana mostly that have lots of cattle ponds and this riparian vegetation. And so why am I talking to you about a mosquito? Like, I'm talking to you about this mosquito, and it's important to everybody because it's a vector for a virus called West Nile virus. And so West Nile virus has neurotoxicological effects on humans and animals alike. And so in very few cases, when people contract this virus, they can have neurotoxicological effects. In fact, it's about 1%. And funnily enough, when I was in between 7th and 8th grade, I actually contracted this virus, and in contracting it, felt extremely sleepy. And so in knowing where this Culex tarsalis exists, we can then predict where people are going to contract West Nile virus and then inform clinics in those areas to actually test for it when people are showing symptoms of having the virus. And so this really is a project that is preventative in nature so that we can monitor where Culex tarsalis exists, inform local clinics so that they can treat West Nile virus with more information and start looking for it because it's not something commonly thought about. And so this Culex tarsalis locality data was collected by students from Carroll College and Montana State University since 2003. So it's been a really long-running project with this sort of preventative idea in mind, in that we make a habitat suitability model, we make a West Nile virus risk map, and then we're able to inform these clinics across Montana. And so I've used a mathematical modeling program called MaxEnt, which stands for maximum entropy, and this uses Bayesian statistics. In this program, you load in several different predictors. So predictors are ecological layers in this sense. So we load in things like precipitation, vegetation, in my case with Ndvi, and land cover, so different categories of land. And they all help train a model using MaxEnt to predict where we're actually going to find this species. And so that's exactly what a habitat suitability model is it creates a map where we can say that based on this locality data and a model made from it, we're likely to find tarsalis in these locations using these predictors or these layers. And so, like I said, I added the Ndvi data, which is a measure of the vegetative index. And so about every 6 hours, a satellite goes over Montana and takes a snap photo that is able to measure the refractive index. That refractive index then is correlated to what type of vegetation is growing in those locations. And so what this layer is telling us is where vegetation is in different types. And then when you compare different months, you can see changes in growth. So you can see when the plains green up, for instance. And so these are two maps that I actually created using Maxent and using several predictors, including these four different months of NDVI data. And so on the left, we have the entire state. And on the right, we have where I've constrained the model to just the Great Plains. And so as you can see, the map on the right, where I constrained to just the Great Plains, it is much more red, which is correlated to higher likelihood that warmer color is a higher likelihood of finding Culex tarsalis. And so if you were to zoom in on this map, there's tiny little pixels that are saying presence or absence. And so Maxent gives you a binary outcome, therefore, that, and in this binary outcome, it's either saying that the Culex tarsalis is present or that it's absent. And so when you have more presences around one another, you get these warmer colors. And so we can kind of get a gradient outlook. And when we compare the same region for the entire state, we see it's a little less warm, it's a little less red. And this is why Dr. Hokett asked me to constrain the model to just the Great Plains. And it's because using Maxent, I basically added a whole bunch of data points where the Rocky Mountain front exists, where there are no tarsalis in this region. And because there are no tarsalis in this region, you make the model look better than it actually does. And so in the outcome, the overall state model gave a better outcome, says that it's a better model, and then when I constrained it to just the Great Plains. But the outcome really isn't a good measure in the entire state because I've added a bunch of absence data. And by adding that absence data, I've improved the outcome of the model, but it's not a true improvement. And so when we look at the Great Plains, we see this warmer color, meaning that you can trap in any of this location in the Great Plains and most likely catch a tarsalis if you trap there often enough, meaning that the tarsalis is ubiquitous throughout this region, that it is all over the place. But this points to the need for, rather than a binary outcome, a more continuous outcome, meaning that at this location, we're 40% of the time going to find tarsalis rather than just, we caught it, it's there sort of outcome. And so because tarsalis is more ubiquitous throughout this region, it actually then makes the model look better because it shows us that we're more likely to find tarsalis in this region. And so when you go in and you look at the specific variables in the model, you have two different ways that you can look at it. The first way is Jackknife variable analysis, which is shown here. And so the top is the entire state and the lower is the just the Great Plains. And this measure is AUC, which is a measure of how good the model is. The top bars here are both the total model AUC. So that's what we want to compare each bar to. The blue bars for each one of the variables is the model without that variable. And then the orange bars is the model with just that variable. And so when you take the variable away, which is what this blue bar is measuring, if you see a big drop, it means that this variable is really adding a lot to the model. And so when you compare the entire state to just the Great Plains, you actually see that there's more sensitivity with these models, more sensitivity to each variable in the model. And so you see bigger drops in the measure of how good the model is, the AUC, when you take the variables away when you're just looking at the Great Plains. You also see that in looking at this, that my September NDVI layer, so the NDVI for the September month averaged, is pretty good on its own, and it drops the model quite a bit from the total model AUC. So this is helping us to identify which variables are really good in the model as predictors and help training the model and making a better model, and also how sensitive the model is to that specific variable. And so the second way to evaluate which variables are doing important things within the model, let's say, is by ranking them based on permutation importance. And so you can see that the September NDVI in both the entire state on the left and the Great Plains on the right is it outperforms the rest of the variables. We see a slight drop between the permutation importance between the entire state and the Great Plains. But like I said, when we look at the whole map, it actually is better at predicting what we've noticed in trapping tarsalis in the Great Plains. So why September? We were really curious about why September was so good at predicting and training this model and predicting where tarsalis exists. And so we looked at then the response curve of this NDVI layer in the model, meaning how does this specific variable interact with the model and whether there's a presence or an absence in each pixel. And so in September, we see a really, really strong positive correlation between the increase in NDVI values, which means that it's greener or has more vegetation there, and actually predicting presence data, which is 1. And the reason why we've kind of deducted in an ecological sense is because tarsalis in September go from being protein feeders, which is blood, and to feed their eggs, to nectar feeding. And so because NDVI is a measure of vegetation and tarsalis start seeking out vegetation in September to nectar feed, that's why you're going to have a positive correlation, meaning that where there's positive NDVI, there's also a presence data for the tarsalis. And so the habitat suitability model was definitely improved by the addition of the NDVI layers, especially September. And then when I constrained the model, it decreased the overall AUC, the overall outcome of the model, but it actually is closer to reality, meaning that we're able to find tarsalis more, and tarsalis is more ubiquitous throughout the Great Plains region, which is what the constrained model shows. This also brings up that Maxent has a weakness in its binary outcome, its presence or absence, and it would be better to have a more continuous outcome so that even though you're going to find tarsalis in the Great Plains, you're going to find it less in certain regions and more, almost all the time in other regions. And so that's where this model really does come into effect in the Great Plains to predict the prevalence that we've noticed of Culex tarsalis. And so I would like to acknowledge all of the undergraduate work that has gone into this program, meaning that students since 2003 have trapped mosquitoes and sorted through thousands and thousands of mosquitoes, hundreds of thousands of mosquitoes to find this specific species so that we can have a locality data set. I would also like to thank Dr. Hokett for helping me learn Maxent, learn ArcGIS, and make this project possible. This project was made possible through funding through Ndvi, so I'd also like to thank them. And thank you all for watching my presentation today, and thank you.